Hey, hi everyone. I think some researchers is joining, but I think it's time, so we will start now. So let me start first, and good afternoon, everyone. Thank you for joining this webinar today. I'm Kyungmin Kim, an associate professor at Yonsei University and host for this webinar as a representative of KSBMR. It is my great pleasure to welcome you all to this valuable joint symposium, the KSBMR, JSBMR, ANZBMS, ISBMR, ICMRS joint webinar titled Innovative Research in MS Musculoskeletal Field. I believe these collaborative events will be a unique opportunity to bring together expert researchers and clinicians from around the world to explore the groundbreaking advance in our field. Our symposium represents the collective effort of 5 renowned societies: the Korean Society for Bone and Mineral Research, the Japanese Society for Bone and Mineral Research, the Australian and New Zealand Bone and Mineral Society, and the Indian Society for Bone and Mineral Research, and the International Chinese Musculoskeletal Research Society. And we are united today by a shared mission, advancing the science and understandings of musculoskeletal health to improve lives globally. Today, we will have one keynote lecture and 4 early and mid-career speakers. And we are honored to host a distinguished lineup of speakers who will share their recent research and valuable insight of this field. And after each talk, we will have a brief Q&A session. And if you have any question, you can type it in the chat box during the talk or use the raise hand feature to ask your question directly after the session. And let me introduce the first speaker, Professor Ji Shan. And Dr. Ji Shan, Dr. Ji Shan is an associate professor in the Department of Orthopedic Surgery at Washington University in St. Louis. He graduated in Nanjing University, and he finished his master's degree at Nanjing University, and he finished his PhD at University of Rochester, and also a postdoctoral degree, University of Rochester. Now he's an associate professor in Department of Orthopedic Surgery at Washington University in St. Louis, and as a cartilage biologist with specialization of osteoarthritis and fracture repair. His research interests span various aspects of bone and cartilage, focusing on the injury, repair, and regeneration of musculoskeletal tissue. He published so many valuable articles in this field, and the title of his talk today is New Insights into Mechanism and Therapy of Fracture Renewal. Professor Ji Shan, please.
Thank you for the kind introduction, Dr. Kim. Let me share my screen. Okay. Can you see my screen?
Yes, we can see. Yeah.
Okay, great.
You can start. Yeah.
Yeah. Give me a second. Okay. So first of all, it's a great honor to give a keynote talk today on behalf of Chinese Musculoskeletal Society. I will talk about a new fracture non-union model we recently developed in the lab and how we use the single-cell RNA-seq approach to explore the mechanism behind the disease. So bone fractures are common musculoskeletal injuries. According to the recent nationwide hospital data, there are about 15 million fractures occur every year in the US, and the incident rate is increasing. Although most of the fracture progress to timely union in underdeveloped regions and countries, up to 15% of the fractures can develop into delayed union and even fracture non-union, which can lead to reduced patient life quality and a significant cost associated with treatment. There are two distinct types of fracture non-union, hypertrophic and atrophic non-union, characterized by different radiographic observations and biological properties. Development of hypertrophic non-union is primarily due to inadequate mechanical stability, leading to excessive fracture callus formation. Therefore, appropriate fixation at the fracture site is usually used to achieve successful outcome in hypertrophic non-union patients. In contrast, atrophic non-union is characterized by limited fracture callus with poor angiogenesis, likely due to the dysregulation of normal biological cues. Treatment of atrophic non-union usually involves complex clinical practice and often requires multiple surgeries. Therefore, atrophic non-union results in significant patient disability and increased cost to the healthcare system. And atrophic non-union remains a major clinical challenge for orthopedic surgeons. Several risk factors have been identified in clinic, including aging, smoking, alcohol drinking, and medical conditions such as obesity and diabetes. All these risk factors actually are associated with chronic inflammation. In vitro studies have demonstrated the inflammation negatively affects the progenitor cell proliferation and differentiation. TNF-alpha transgenic mice and collagen-induced rheumatoid arthritis mice displayed reduced callus formation and impaired fracture healing. So to tackle this inflammation, anti-cytokine therapies have been used in animal models and human patients. Preclinical animal studies showed a positive effect of the TNF-alpha inhibitor on restoration of callus formation and biomechanical properties of fractured bone in wild-type rats under inflammatory conditions. However, a human cohort study demonstrated a negative long-term effect of TNF-alpha inhibitor treatment on fracture healing. This evidence implicates that simply inflammation blockage is not a good treatment option for delayed union and non-union. Therefore, there is an urgent need to develop mechanism-based therapies. To study the mechanism behind the disease, several animal models have been developed. Critical size bone defect is one of the widely used model. This model requires a special set of tools to remove the fractured bone to create critical size bone defect. The model has high clinical relevance since it mimics osteotomy surgery. However, the procedure is expensive and relatively complicated. The other model is a periosteum stripping model. A relatively smaller fracture gap can be created in combination with removal of periosteum tissue. The mice will establish the atrophic non-union around 18 weeks after surgery. Both of these two models are useful to test the efficacy of the biomaterials. However, it is not very useful or very helpful to explore the mechanism because the local tissue is simply removed. We cannot really understand how this tissue responds to the pathological conditions. Therefore, we decided to develop an inflammation-mediated fracture model in the lab, which will give us the opportunity to delineate the mechanism. We collaborated with Dr. Abu Ammar in our university and used the KBXN-induced rheumatoid arthritis as a systemic inflammation model. We IP injected KBXN serum every 5 days to induce and maintain the elevated inflammation. Consistent with expected increase in inflammation, we confirmed that RMIs displayed significantly higher luciferase activity in NF-kappa B reporter mice, especially in limbs compared to the PBS group. More importantly, the systemic inflammation can be maintained in an elevated level at least up to two weeks. Under this inflammation condition, we found RMIs displayed no cartilage and no new bone formation by 10 days post-fracture. In contrast, the broken bone in control mice is perfectly bridged by cartilage tissue and a newly formed bone. One more finding from histology I want to emphasize is that morphologically, the tissue here, the tissue in the gap between the broken bones in the RA mice looks like fibrotic tissue. And this tissue persisted throughout the fracture repair process in RA mice, which I will show you later. Since we know that in our tibia fracture model, angiogenesis peaks around day 10, so we performed the micro-CT scanning to examine the angiogenesis at the day 10 post-fracture. Micro-CT data confirmed that inflammation substantially reduced angiogenesis in RA mice, indicating that inflammation has adverse effect on the blood vessel restoration. We also examined the bony callus formation by using micro-CT. By day 21, bony callus formed and unified broken bones already in the PBS group. However, almost no bony callus could be found in RA callus, and the fracture line, here, is persisted until day 21 based on the micro-CT image. More importantly, instead of newly formed bone, type 3 collagen positive fibrotic scar tissue still filled the gap between the two broken bones in RA mice by day 21. And as expected, the fibroblastic scar tissue leads to remarkably poor mechanical performance in RA mice at day 28. RA fractures displayed significantly lower bone strength compared to the control fracture. Here I also included two more pictures. We can see here, this is the tibia, fractured tibia after 180-degree rotation. The fractured bone in RA mice was still connected, which means there is something, there is a tissue in between, which we further confirmed by histology. This is a soft tissue. So altogether, the RA mice displayed no fracture callus formation, diminished angiogenesis, fibrotic scar tissue within the fracture, and poor mechanical performance, all of which are consistent with clinical manifestations of the atrophic non-union patients. Therefore, this work highlights a novel RA non-union model with high clinical relevance and provides a useful tool to study the pathology of the atrophic non-union under inflammation particularly. To explore the cellular and molecular alterations associated with fracture, we performed single-cell RNA-seq. We isolated fracture callus tissue along the periosteum surface to minimize unnecessary contamination of the original bone cells and the bone marrow cells. 20 distinct cellular clusters were observed. Specifically, 3 major populations were identified: CD68 positive myeloid lineage, PRX1 positive mesenchymal lineage, and CD31 positive endothelial cells. When we review the overall cellular compositions in fracture callus, I think it's quite clear the alterations between the control and RA groups, and also in different time points. I mean, day 4 and day 7 obvious, in line with the elevated systemic inflammation. We identified higher proliferation of inflammatory endothelial cells, T cells in RA mice. Although the inflammatory MR macrophage was slightly decreased, but in contrast, a reduction in anti-inflammatory M2 macrophage was also observed, which this M2 macrophage plays a critical role in resolving the inflammation. So that means the overall fracture environment prone to the inflammatory conditions. We then focused on the population of mesenchymal lineage because that's the cell plays a key role in forming fracture callus. We further defined the 5 distinct cell types according to the lineage-specific gene signatures, which I'm presenting here. For example, common mesenchymal cells, class 13, committed mesenchymal progenitors, class 1, osteochondral progenitors, class 15. This is a very interesting cluster because the both SOX9 and SP7 was presented. Class number 2 and 4 basically are fibroblastic tissue or fibroblastic progenitors because they are highly express some of the fibrotic tissue marker genes. So we next examined how the progenitor cell proliferated and differentiated in different conditions, normal condition or inflammatory condition. So from this proliferation cell fraction picture, I think we can easily distinguish under the normal condition from day 4 to day 7, the progenitor cells, I would say the normal skeletal repair cells, including class 13, 1, and 15, was largely expanded. At the same time, the fibrotic like cells or progenitor cells was reduced. However, under the RA condition or under inflammation condition, so the fibroblastic tissue progenitor cells was largely expanded. This observation actually is consistent with our histological findings that the fibrotic tissue formation in RA fracture. Regarding progenitor cell differentiation, we applied the trajectory assay. Interestingly, the common mesenchymal progenitor cell, the class 13, predominantly occupy the one trajectory, and basically it's the center or the start of the trajectory. And the osteochondral progenitors, class 15, and the fibroblastic precursors, class 2, as well as the fibroblast, were exclusively located along the opposite branches. This distribution likely means the distinct path of the either skeletal or fibroblastic differentiation. More importantly, the cell from the control fractures, which is the unshaded portion, were prone to follow the skeletal differentiation pathway. However, the cells from the RA fractures, which is the shaded portion, exhibited a greater portion within the fibroblastic trajectory. This observation suggests that systemic inflammation impacts progenitor cell proliferation and differentiation. Lastly, we performed pathway analysis and revealed that the RBPJ-kappa-mediated Notch pathway as the most upregulated pathway in progenitor cells, suggesting that it's potential role in dysregulating mesenchymal differentiation under RA conditions. So here I just give a example how we use the single cell RNA-seq data to explore the mechanism in the cell lineages of our interest. So basically, you can get a lot more data or a lot more information from this data set. So as I emphasized, RPPG kappa notch pathway as a potential downstream. Next, we used a genetic approach, genetic animal models to try to provide a more focused understanding of the precise mechanism in progenitor cells. So to achieve that, we generated the PRX1 CRE-ERT2 RKK2CA mice. So PRX1 is which can target let us specifically target the progenitor cell lineage, and IKK2 is a key transcription factor mediating the NF-kappa B or inflammation pathway. So using this PRX1 CRE-ERT2 RKK2CA mice, we can specifically activate intrinsic inflammatory response in mesenchymal progenitors. Histological evaluations of the fracture repair were performed using the Austrian blue hematoxylin. So the control mice basically exhibited normal healing process with robust cartilaginous and bony callus formation in either day 10 or day 14. However, this IKK2CA activation mice or the means the mesenchymal cell has the higher inflammation. The mice developed impaired healing with less amount of the cartilage tissue and a significantly reduced newly formed bone in either day 10 or day 14. We also performed the micro-CT scanning, scanning to review the bony callus formation. The micro-CT imaging from the control mice displayed a nearly complete region of the bony callus by day 14. In contrast, IKK2CA mice presented less bony callus, newly formed bony callus formation, and the fracture line was persisted. Collectively, this data strongly suggests that IKK2CA expression disrupts the fracture repair process by disrupting the progenitor cell differentiation potentially. So to further conform RPPG kappa mediated notch as the potential downstream of inflammation in progenitor cell lineages, we generated PRX1 CRE-ERT2 RKK2CA and RPPG kappa loss of function mice. So basically, please pay more attention on these two groups I highlighted in red box. So again, the IKK2CA mice clearly show under inflammatory conditions, cell differentiation was impaired, fracture healing was impaired. However, when we delete the RPPG kappa gene under this kind of IKK2CA background, we can see the cartilage tissue or the chondrogenic differentiation in cells can be partially restored, and a relatively normal bony callus can be formed in this kind of RKK2CA, RPPG kappa loss of function mice. This in vivo evidence indicate that RPPG kappa is one of the potential downstream target of inflammation in progenitor cells. Again, we performed the micro CT to review the bony callus formation. When we compare the IKK2CA group with IKK2CA RPPG kappa loss of function group, we can clearly see a relative normal bony callus can be formed by loss of RPPG kappa in progenitor cells, even under this kind of IKK2CA RKK2CA background. So due to the time limit, I will skip the mechanism how we started the epigenetics in this project. I will give a quick summarize here. So basically, we created a novel clinical relevant fracture non-union model. That means the elevated inflammation can lead to fracture non-union in mice. And then we using single-cell RNA-seq approach in combination with genetic animal models and established an NF-kappa B, DMT3B, which is epigenetic factor, RPPT kappa, the pathway in regulation of progenitor cell differentiation, which can contribute to fracture repair process. I hope this work provides some new mechanistic insights and therapeutic targets for fracture non-union, particularly associated with systemic inflammation dysfunction. So I already summarized, we can skip here. Finally, I want to thank all the lab members who contribute to this project. I also want to acknowledge my collaborators. Yusuf is the person contribute to the KBXN RA model. Regis Foch help with the animal model and some of the in vitro studies. Bo is the person who helped with the bioinformatics and analyze. And here is my funding resources from NIH and also from our department. With that, I want to thank you all for your attention, and I can answer any questions. Thank you.
Thank you, Professor Chen. It's great talk. It's so interesting. And is there any comments or question from the audience? As I mentioned, you can use chat box, or you can just enable your camera and mic, and you can ask directly. So, okay, actually, Professor Chen, I have one simple question.
Sure.
Actually, in clinical field, one of the most strong anti-inflammatory medication is steroid. So, but it's very well known to steroids. It's not that good enough, not that good for bone and cartilage as well. But according to your research, the anti-inflammation therapy can affect beneficially on fracture healing, kind of on union. So is there any effect steroid treatments in suppression inflammation? And is there any evidence it can upregulate RBPJ kappa when you using steroid?
Yeah, I think, first of all, I personally don't think anti-inflammation is a good option for patient treatment. As you mentioned, because first of all, it's very difficult to locate a precise window for this treatment, because early on hematoma stage or early inflammation is good for fracture healing. They can attract or bring the cells, right, especially the progenitor cells to the injury site, which can later on contribute to the callus formation and the fracture healing process. So clearly, if you want to use this kind of anti-inflammatory treatment, you have to avoid this early window. However, in some of the patient, as I mentioned, for example, diabetes patient or smoking or alcohol drinking patient, they already have a elevated systemic inflammation there. All right, this endogenous inflammation from the very beginning, not only I would say they may can help attract the cells, but they already damage or impact the endogenous progenitor cells. So that's why when those patients experience fracture, it will eventually have a delayed healing or even non-union, right? So that's what I think directly target inflammation is not a good option. We need to understand what's the downstream target, like RBPG kappa. So in that way, we can target RBPG kappa or notch pathway to develop new drugs or new medication to treat those patients. So which I don't have time to show as we epigenetically modify the cells, the progenitor cells, and then I use the biomaterial, put the cell back to this RNA mice. Actually, those mice can be healed. The fracture can be healed, relative, normal. So that gives us some confidence, if we can dissect the mechanism and use and provide a mechanism-based therapy that will achieve eventually a successful outcome. So yeah, that's what I can say now.
Another option can be, I mean, the therapeutic, it can have some data about it affect the differentiation of stem cells and also beneficially affect the healing. So is there any evidence that therapeutic treatments also affect your pathway?
So I will say PTH is not, stand out, let's say in this way, in our pathway analysis. So that's why in this study, I'm not focusing on the PTH pathway, but I agree with you, in vitro study, right? Especially PTH showing, actually that's the work from our group. We show PTH can accelerate fracture healing in Asian mice. So I will say they still have some effect promoting progenitor cell differentiation and try to balance osteoblast differentiation and osteoclast function, right? So it's also another key topic, how this kind of balance can eventually help fracture bone back to the normal shape and again the mechanical property. So it's PTH. Yeah, but again, that will not be a mechanism-based therapy, right? It will just, PTH have this kind of broad effect. They can have some osteoblast anabolic effect. So as a potential treatment. So yeah.
Thank you for raising. I think we have to move the next talk.
Sure.
So thank you for giving a great lecture. And let me introduce the second speaker, the Sang-Uk Jo, and he finished his master's bachelor's degree about information and management in Peking University, China. He also finished his master degree in data science, Peking University, China, and also he recently finished his PhD in biomedical engineering in Yonsei University in South Korea. And his major research area is AI in medical images and osteoporosis, sarcopenia, and web-based software developed. He awarded so many great awards from Korea and other international society and published the paper regarding AI research in detecting fracture and predicting the fracture as well. Now, today the topic is really interesting. Is the title of his talk is leveraging ECG data, improving fracture risk assessment from reduced consortium. Sang-Jo, please.
Yeah, thanks for introduction. Can you see my screen now?
Yes, I can see.
Okay. Good afternoon, everyone. My name is Sang-Uk Jo, researcher at Yonsei University College of Medicine. It is really great honor to present our recent work in this webinar. Today, I will be presenting presenting on the topic, leveraging electrocardiogram data to improve fracture risk prediction, LIDAS consortium. I will provide further detail as we progress through the presentation. I have no disclosure, and Korea is about to enter a super-aged society by early 2025, marking a significant shift in public health challenges. This change underscores the critical need to transition medical service from disease treatment to proactive prevention. Fracture are a major concern in aging population, and globally, a fracture occurs every 3 seconds, and incidence rate are rising. With appropriate intervention, fracture rate can be reduced by 50% to 80%. So similarly, cardiovascular disease remain a significant health concern in Korea, ranking as the second leading cause of the death in 2023. Also, prevalence of the heart disease increased every year. Musculoskeletal and cardiovascular diseases show clear age-dependent pattern. Many researchers indicate that this condition influence each other, with aging playing a key role. A study published in JAMA in 2009 highlighted the association between ECG abnormalities and hip fracture, such as a heart failure and ischemic heart disease. Some additional studies have linked condition identifiable through ECG, like atrial fibrillation AFib, to fracture risk. Recent research has some leverage ECG signal data to identify biomarker of aging. So notable examples include the Mayo Clinic studies, the Brazil code cohort, and Taiwan ECG 12net project. These studies have demonstrated that discrepancy between chronological age and ECG estimated age are strongly associated with cardiovascular disease and mortality risk. However, despite evidence of an association between cardiovascular and musculoskeletal health, there is a lack of the research exploring this relationship due to data limitation. Our study aimed to investigate the association between ECG abnormalities and incident fracture. Also, we want to examine the relationship between ECG estimate age and the fracture risk. One significant barrier to advancing research in this area is the complexity of unstructured medical data. Frequently used diagnostic tools like a spine X-ray and ECG hold a high potential for primary care, but face challenges such as requiring significant time and effort from the expert, and limited scalability beyond the label dataset, and restricted utilization of the unstructured data. To address these challenges, we launched the Big Data Smart Curation Technology Development Project in 2022, supported by the Korean Health Industry Development Institute. The Leave Your Data to Us consortium, we shorten as the LIDAS, we comprising 4 institutions here and aim to enhance the medical data usability, improve the data labeling efficiency, and develop the guideline for high-quality medical data set. Our team at Severance Hospital, we established the BERT-X cohort, which includes the data from individual with the spine X-ray and ECG and clinical variables. And using this data set, we developed and published a model for detecting fracture and osteoporosis based on X-ray data. And also we released open source software for pre-processing and curating X-ray and ECG data. So if you scan this QR code, you can visit our website. And we In this study, we used the outpatient-based cohort as a derivation set who underwent baseline X-ray and follow-up scans, along with the 12-lead ECG at Severance Hospital from 2007 to 2018. A total 6,986 patients were constructed. Primary outcome was the incident fracture event, including the morphologic or clinical vertebral fracture and non-vertebral fracture at hip, wrist, upper arm, and other position, and certain by the medical record and imaging data. For the ECG dataset, we matched the classification framework of the publicly available PTB-XL dataset with the GMUS data and underwent the confirmation process by cardiology experts. The ECG-based disease classification system is structured into 5 main superclasses, including categories such as the normal, conduction disturbance, and more hypertrophy, and so on. And the video on the left is the tool we developed to confirm the label for ECG data. All data reviewed by the cardiology expert. And right image is an example of the ECG waveform data analysis and showing how various duration and parameter are extracted. The ECG estimated model was developed by one of our consortium member, Professor Seung-Chan Yu, using a CNN-based network. This model was validated across the dataset, including the UK Biobank, SHC, and other big ECG cohort. So this model and related research was presented at the American Heart Association 2024 conference. During a median follow-up of the 5.4 years, 11.2% of the participants experienced an incident fracture. The median age was the 65.7 years with the 60% female participant. The data analysis was conducted based on the test set, which was not used in the development of the aging model. So in our model, our analysis comparing the chronological age and ECG estimated age and demonstrated an R-square value of the almost 0.3 and mean absolute error of the approximately 5.5 years. In our analysis of the association between ECG labeled abnormalities and incident fracture, we observed the AFib, sinus tachycardia, longer corrected QT duration were all significantly associated with the higher risk of the incident fractures. This association remained robust in multivariable model adjust for age, sex, and previous fracture history. In contrast, hypertrophy and non-ischemic ST changes were not significantly associated. We used the multivariable linear regression model to explore factor of the contributing to difference between the ECG age and chronological age in the test set, especially presence of the AFib and morphological vertebral fracture increased the predicted age by 2.74 years and 1.74 years respectively. Participants were divided into 4 groups based on the chronological age and the presence of the accelerated ECG age. Here's the 4 group here. And compared to the reference group, the group 1, 2, 3 showed the higher proportion of the osteoporosis and prevalent fracture and instant fracture, also myocardial infarction and AFib. The proportion is higher than the reference group. A Kaplan-Meier curve for instant fracture demonstrated that while the reference group and the group 1 were not statistically significant, but group 2 and group 3 showed a significantly higher fracture risk. Specifically, the fracture risk was the 80% higher in group 2 and 180% higher in group 3 compared to the reference group. So further analysis using the Koch's proportional hazard model confirmed that the difference between the ECG predicted age and chronological age was independently associated with the fracture risk. This remains significant even after adjust for age, sex, prevalent fracture, and osteoporosis. And for each one standard deviation increase in predicted age difference, the fracture risk significantly increased also. So overall, we found that AFib and sinus tachycardia, QT corrected duration were all independently associated with the incident fracture, even after adjusting for age, sex, and previous fracture history. And furthermore, 40% of the variance in predicted age difference was explained by the chronological age, AFib, QT corrected duration, and morphological vertebral fracture. In this study, we show a proof of concept that ECG predicted age show a potential to explain the fracture risk. Here's the picture from the Schurer's work, and Schurer and her colleagues explore the potential mechanism linking AFib to fracture and suggesting that the AFib could independently contribute to reduce the bone mineral density. Similarly, in our study, showed that AFib was the more prevalent in the fracture cases and significant influence the ECG predicted age. However, even after the adjustment in survival model, the association between the predicted age difference and fracture remained independent, suggesting that factor beyond the AFib maybe also contribute this relationship. Additionally, other studies have suggested various mechanisms involving the nervous system. For instance, ECG age may reflect the aspect of the physical frailty and autonomic nervous system aging, which are associated with the increased fall risk and accelerate the bone loss. This finding indicate that ECG age could serve as a marker for this underlying weakness, although further research is needed to validate these connections. To gain more comprehensive understanding of the aging, future research should explore multi-dimensional approaches. One promising detection is the development of the fusion model that integrate the spine X-ray and ECG data to predict the aging-related health outcome. Despite its strengths, our study shows has several limitations. The data set were limited to Korean individual requiring validation in our other population. Individual under the 50 were excluded in this study and limiting the generability to younger adults, and measure of the frailty and physical performance are not unavailable, and leaving gap in the understanding spine age and frailty. In summary, our study demonstrated that the AFib and QT corrected duration and sinus tachycardia were independently associated with the fracture risk, and ECG age estimated from the waveform data predicted fracture risk independently of the chronological age, osteoporosis, and other covariates. This finding highlight the potential of the ECG data to enhance the predictive model for aging and fracture-related diseases. And yeah, I finished my presentation, and I would like thank you to Professor Nam-gi Hong and Kyungbin Kim and Hitae-yu and Seung-chan Yu for their guidance. And I also extend my gratitude to Leidos Consortium and the Bone and Mineral Research Team at Severance Hospital for their support. And thank you for your listening.
Thank you, Sang-Cho. It is really great talk, interest topic regarding ECG can predict association of fractures. So is there any question and comments from the audience? So because it's already we ran behind the schedule, so I will give a very brief questions. And actually, in your data set, you included several different disease category like MI or arrhythmia. So some disease from like a rhythm-based disease, or some just to the anatomical disease or vascular disease. So in your analysis, is there any difference according to their baseline heart disease? Because some shows very distinct ECG patterns or some are not. So I mean, is there any association can be different according to their baseline disease?
Yeah, thanks for your question. I think it's a really good question. In like the classification system, we use the PTBXL. They already separate the diagnosis and rhythm and statement. And we try to check the relationship between those different the label. And we show the AFib and other diseases showed which we selected is to show the high statistical relationship between that labels, and the other label we cannot show some significant like relationship between label and so we exclude that kind of that label.
One question from the audience, and Dr. Anderson asked about what are the next steps with this work? Is there any way to implement this finding to clinically?
Oh, yeah, thanks for your question. And yeah, of course, like because we found some like if a patient have AFib or other related diseases, or we can more focus on those patients whether they will have some fracture. So we will try to build some model to understand the aging first, and after that we will try to find some way to implement implement some those methods in the clinical session or clinical setting.
Okay, thank you, Sang-Cho. And now we will move the next talk. And from next talk, we'll moderated by the Professor Mizuguchi from JSBMR. So Professor Mizuguchi,
Okay.
please.
Okay, good afternoon, everyone. My name is Toshihide Mizuguchi. I'm from Tokyo Dental College. So I'd like to introduce the next speakers. We invited Dr. Hiroyuki Okada as a speaker from JSBMR. He is an assistant professor at the Center of Disease Biology and Integrative Medicine, the Graduate School of Medicine at the University of Tokyo. He is one of the leading young researchers in our society, and he recently originally developed single-cell analysis technology. His title is Bone Deep Insights from Single-Cell and Subcellular Special Transcriptome Mix. Okay, Dr. Okada, when you're ready, please start your presentation.
Thank you for your kind introduction. Can you hear me?
Yes.
Okay, so thanks organizer for giving me the opportunity to talk about my latest work. I'd like to share the latest transcriptomic approach. First of all, let me introduce myself. I am an orthopedic trauma surgeon. in the aged society like Japan, the number of hip fracture patients is increasing. So this is a social burden. So our motivation of my translational research is how to reduce the number of hip fracture patients. To reduce the number of hip fracture patients, it is very important to understand bone remodeling process deeply, because disrupt of bone remodeling process causes osteoporosis. So recently we can use the single-cell RNA-seq. So we want to depict the multiple cellular system like bone remodeling process. We'd like to apply the single-cell RNA-seq for bone remodeling process. We can use it to clarify the heterogeneity in cell suspension, but it's very important to apply for bone remodeling process, because there is a big limitation. The diameter of cells going through the microfluidic device is less than 70 micrometer. So we cannot separate osteoclast at single cell resolution. So we have built another technology to clarify the osteoclast gene expression profile at single cell resolution like this. We call this platform as intra-single cell sequencing, ISCSEC. This is a very simple protocol. We took pictures with confocal microscope. Then we adjust the depth position of tip of the probe and sample any cellular components that we wanted. And then finally, we can construct the cDNA library for RNA-seq. So these videos are when sampling the nucleus from living osteoclasts. So using this new novel technique, we can illustrate the cellular, subcellular heterogeneity inside living osteoclasts. And the important thing, the more striking thing is that the majority of the large osteoclasts were not differentiated components. These results are very, we are very amazed with these results. And I am also a bioinformatician, and we wrote educational review paper last year and titled Practical Compass of Single-Cell RNA-seq. And in this review paper, I explained about how to build the atlas level single-cell RNA-seq like this. So if you are interested, please check this paper. So today's topic is the bone-forming cells like osteoblast and osteocyte. In the textbook, this type of picture is usually used to depict the osteoblast to osteocyte lineage. Mesenchymal stem cells differentiate into the osteoblast and osteocyte. And the osteocyte's position in this lineage is always in the final part of this lineage. However, we do not have quantitative evidence to depict whole lineage of mesenchymal stem cell to osteocyte. So I'd like to depict this lineage, whole lineage, with new technologies. With single-cell transcriptomic, we can get markers, lineages, and functions simultaneously. This is a big advantage using single-cell transcriptome. However, we cannot get the localization information with single-cell transcriptome. So spatial transcriptome is a promising tool to add the localization information onto the single-cell transcriptome. So I'd like to propose this combination is very powerful tool to illustrate the bone remodeling process. So there are two modalities of spatial transcriptomics, and the Visium has five-year history. And Visium is the sequence-based technique, and theoretically we can get a whole transcriptome. However, the RNA quality is problematic. When dissociating process, RNA is always deteriorated. And many researchers try Visium, but they cannot illustrate bone remodeling process. So we'd like to try another technique of spatial transcriptomics. 10x genome inside you. This is the imaging-based technique. The number of genes detected were fewer, but the resolution is higher, much higher compared with the Visium. And we hope that the degraded RNA is torrent, maybe torrent for the new type of spatial transcriptomics. So we applied 10x genome for bone. And first, we built a single-cell bone atlas with only public dataset. There are plenty of public datasets available, so we do not have to perform our own single-cell experiment. And we included single-cell datasets other than 10x Chromium. This is important to reduce the biases, technical bias. And this is the result of our bone single-cell atlas. This is mouse version. And we can collect 72 kilo cells, and we can divide into 20 cell types. And we can get the osteoblast and osteocyte lineage. And the osteocyte were positioned in the last part of osteoblast lineage. So we call this osteocyte classical osteocyte. However, there is a big problem because this classical osteocyte did not express higher expression of rank ligand. In the common sense, osteocyte secretes rank ligand. However, this osteocyte did not. So we want to depict another subset of osteocyte inside osteoblast big island. We can depict the osteocyte enriched region. So we call this region as alternative osteocyte. And alternative osteocyte secretes rank ligand. And the function is diverted between two subsets of osteocyte. Classical osteocyte were specialized in bone mineralization. In contrast, alternative osteocytes have the function of bone remodeling because alternative osteocytes secretes rank ligand. So this is a short summary of two subsets of osteocyte, classical osteocyte and alternative osteocyte. Sclerostin is commonly expressed in both two subsets, and SB7 and RANK2 expression were diverted. This is very amazing result. And the alternative one secretes rank ligand, and they are specialized in bone remodeling process. So with using the single-cell atlas, we can illustrate two subsets of osteocytes. So next step, we have to confirm this existence. So we apply spatial transcriptome for bone as this. And there are some tricks to avoid RNA degradation. The most important thing is that to minimize the decalcification time, because decalcification causes the RNA degradation. So we minimize the time within the 72 hours. And we made the 5 micrometer slice under RNA-free condition. And we made this type of slice on the specialized slide. And we can discuss the RNA expression profiles on the same slide. And this is a big advantage of the spatial transcriptome. And we use the DNU version 1 adding on the 100 custom gene panel. So this is the result. We can discuss the 47 genes on 17 kilo cells. And we can discuss about the RANK2 and SP7 dominancy. So the important thing is that the intersection part of RANK2 and SP7 is minority. So we can divide the RANK2 and SP7 dominancy. And this is a raw result of RANK2 and SP7 positive cells. And this is very sad, and we cannot illustrate the localization divergence, trabecular and cortical. So next, we transfer the labels of the classical and alternative osteocyte profiles onto the same slide, and we can depict the different localization of two subsets. The important thing is the alternative osteocytes were only in trabecular bone, where bone remodeling process actively occurs. So there is a big limitation because 10x genomics did not provide our specialized cell segmentation tool. There are many gene expression profile signals inside bone. However, we cannot recognize cells inside bone. So we are now developing original cell segmentation tool using the cutting edge deep learning technology with manual segmentation, and we can maximize the number of cells inside bone. So we can discuss, next stage, we can discuss the osteocyte gene expression profile. So this is the first trial of the combinatorial approach of single cell atlas with the spatial transcriptome for bone. And we can identify two subsets of osteocyte. One is classical and the other is alternative one with quantitative evidence. So we are now developing the new model of bone remodeling process with quantitative evidence to pursue the new paradigm in bone remodeling. So next week, I will talk at Cell Bio in San Diego. So if you join, please discuss with me in person. So this is the acknowledgement of my research. Many researchers collaborated. I collaborated with many researchers. Thank you for listening. I'm happy to take any questions. Thank you.
Thank you very much for your great talk, Dr. Okada. Now, floor is open for discussion. Are there any question or comment? Dr. Okada, you find the two kinds of the osteocyte, right?
Hi.
One is classical osteocyte and another one is alternative osteocyte.
Yes.
So I'd like to want to know the distribution pattern of these cells.
So the crude analysis, I focused on the cortical and trabecular, and in the raw analysis, the alternative osteocyte will only exist in the trabecular bone.
Oh,
Yeah. So the function is completely linked.
I see. Which
Yeah.
cell express lung ligand?
So alternative one.
Alternative one.
Yes.
I see. And how about the OPG?
OPG is expressed in both subsets.
I see. Okay, the lung ligand just only express alternative one and then both are.
Yes.
Okay, so are there any question? Shane, Dr. Shane?
Yeah, may I have a quick question? So,
Yeah.
because I'm also working on this field, I know it's very hard to digest and release the mature osteocyte, right? Because those cells are embedded in the matrix. So I'm just wondering how confident you are to define those cells as either classical osteocyte or alternative osteocyte, because so far, based on what I talk to others, they all agree it's quite difficult to release osteocyte.
Yeah, I agree with you. So the point is the power of many. So to increase the number of cells of bone, so the percentage of osteocytes is the same, but we can discuss 100 osteocytes with 72 kilo cells inside. So the power is very important. Yes.
Yeah, I agree. You can use the high throughput technology and to push as many cells as possible for the single cell RNA sequencing. But I still have the concern, how easy you can release the cells, or I mean the osteocyte from the embedded matrix. So yeah, but a beautiful talk, and
Yeah.
look into your future work.
Yeah. So to confirm how difficult it is, so I'd like to investigate more about the spatial transcriptome. So
Yeah.
because we can get the localization information with spatial transcriptome. So
Sure, yeah, that's another powerful tool. Yeah.
yes.
Thank you.
Thank you.
Okay, does anyone have another question? Okay. So, okay, so I just a quick question. You improve the RNA quality, right? So, and the decalcification time is very short, say just only 72 hours. How about the fixation time?
Yeah, so from my knowledge, 72 hours, same.
Fix 72 hours?
Yeah, yes,
And the decalcified 72 hours.
yes,
Okay,
yes.
I understand. Okay, does anyone have another question? Okay, okay. Okay, time is up. Okay, thank you very much for your great talk, Dr. Okada. So we'd like to move on to next speaker. Thanks.
Thank you.
Dr. Anderson will moderate next remaining two lectures. So Dr. Anderson, please.
Yeah, of course. Thank you, Dr. Kim. My name is Dr. Cara Anderson. I'm the outgoing acting co-chair of the Australian New Zealand Bone and Mineral Society Early Career Investigator Committee, and I'm pleased today to be introducing the final two speakers. Dr. Polpa Durumban is an early career researcher in the Department of Biomedical Engineering at the University of Melbourne. He's skilled at applying software development, machine learning, and high-performance computing techniques to medical signal and image analysis for clinical applications. And he enjoys working with various medical imaging modalities. His current research focuses on developing image analysis and modeling tools for assessing structural degradation of musculoskeletal tissues with arthritis using high-resolution computer tomography datasets. The tools he developed are currently being used at the University of Melbourne and by collaborators around the world, and his talk is entitled Imaging the Osteochondral Unit: Insights from the Synchrotron. Thanks.
Nice. Thanks, Kara, for the kind introduction. I hope my audio is working. Please let me know if it's not. I will share my screen promptly. This should be the one. So, all righty. Okay. So, hi, everybody again. Old and new friends. I've noticed a few familiar friends in the audience, so it's great to see you again. So today I'll be talking more about the imaging side because seems like we have a lot of good mix of basic translational biochemicals, all the omics study, which is very interesting. So today I want to present another aspect that we can implement with a focus on the tools rather than the biology that we can quantify, but rather I would like you, the audience, during this talk to keep asking yourself, how can you integrate maybe this kind of imaging modality into your work and the questions you're trying to investigate. So first of all is the osteochondral unit. So bone, calcified cartilage, and cartilage plays a critical role in maintaining joint mobility and normal load transfer. Recent evidence shows that these tissues undergo tightly linked remodeling activities together as a single osteochondral unit. So, as we all know, disruptions to these remodeling activities can lead to joint diseases such as rheumatoid arthritis, osteoarthritis, many of which has listed all of these pathological features here. So, with the latest consensus, if I can say, that bone and cartilage remodels together rather than as separate tissues. What our team is interested in here in Melbourne is that this little unit as the osteochondral and at where the cartilage and bone meets each other, which is the osteochondral interface. So how do we actually capture this osteochondral interface? So obviously the traditional way to do it and the gold standard for the moment is to actually do histology, where you can see here that you can see, at the top layer, the non-calcified cartilage, NCC, or we can also see CZZ, calcified cartilage zones, and then SB here, subchondral bone region. Easy enough to see in histology, not so easy to see in 3D with micro CT. So to visualize, so CT is great in visualizing mineral component in 3D. However, due to the small size, as many of us work in preclinical models like mice and rats. This is seeing the osteochondral interfaces often beyond the resolution that is available to our regular systems. As in this example of a mouse micro-CT scans in vivo, we can barely see the delineation of the interface, let alone the separation between the subchondral bone and the calcified cartilage region. So we need some tricks to make it work. And here is where I would like to introduce synchrotron imaging. So a synchrotron is a type of particle accelerator that produces extremely intense and highly focused beam of light, ranging from, you name it, it's like you can get infrared to X-ray. However, as opposed to the particle collider, the one everyone talks about at CERN, synchrotron imaging uses these particles to shoot into objects to get images. So you can see here there is composed of two rings, and in the inside of the inner ring is the one where the particle gets accelerated. And once it's accelerated with enough energy, it then gets stored at the outer rings. These lights can be up to many million times more brighter than the sun, so obviously not for the naked eye, but it's pretty good at visualizing things in fine details. So, okay, it's cool and all. This thing is the size of a football court. So obviously we can't fit that in our own lab. There is a number of facilities that is available near you, hopefully. There are a number of facilities. Most of these are located currently in Japan and China. Korea has one facility, Thailand has one, India has two, and of course in our little pocket here down under, we have one in Melbourne as well. So these are generally run by the government agencies. So don't feel discouraged, and access to these facilities are generally very easy. You might need to apply to get access and beam time, and these are generally free. So please just feel free to craft a proposal, contact your local facility when they have rounds open. The one in Melbourne opens twice a year or more, and it's free. And there are beamline scientists and a physicist who will be helpful to help you set up the experiment to acquire what you need. So technically you don't need to be an imaging expert to actually get these kind of images as I'm about to show you. Pretty accessible, sounds really cool, sounds out of reach, but it's actually not. So with synchrotron radiation micro-CT, we can get one monochromatic beam. So it's better than your tabletop lab micro-CT systems where it is better at acquiring different layers of minerals. It can get to nanoscale resolution. It has higher signal-to-noise ratio, and as you can see in this comparison, regular micro-CT image can be shown on the left, and this is the best the system can do. And the right here is where we use space contrast synchrotron CT, where you can actually see that low mineral region sitting on top in the joint space area. And in terms of mineral density, I can show you that it is great, as my friend here has been featured on the cover of JSBMR a few years back. Here we can get the metabolic activity of the bone, where you can see the layers in orange showing the abnormal metabolic activity of the bone around the lesion area. And we can only get this level of contrast between normal metabolic activity and abnormal activity with synchrotron monochromatic beam. So this is just going to be like a demo study that I ran recently, just to show you what it can do and how to process the images. So in terms of this data set, we have a mouse model of OA. So these are spontaneous models, so it has to grow really old. Then we dunk them in contrast agents because we also want to see cartilage, and so we can quantify the remodeling activity together. Then we place them in the humidity chamber to keep the cartilage humid, otherwise it's going to dry out during the imaging process. And then we chuck them into this high energy beamline. The images, despite me being all nice and excited about Asia Pacific beamlines, this one was actually acquired in Switzerland. Just so you know, but it pretty much works the same way. So the processing approach, I did take a very similar approach to this nesting doll approach. So we segment one layers at a time. So we go from, this is the image that we can get. Then we can start to segment the cartilage out because that's the first thing we can easily remove. Then the second layer is to actually just get the bone from which we can define the cortical region of the trabecular to separate the trabecular region from the cortical region. And in this cortical region, you can see that we can use this monochromatic beam to separate the low mineralized region from the high mineralized region within this cortical area. Which, now that we have successfully segmented all these cool regions, we can then apply the usual, morphometric measures like the ratio of low to high mineralized tissue, porosity, thickness, and so on. So just to show you an exemplar visualization of what we can get, for example. So in this data set that we have acquired, we have taken it with 3 micrometer voxel size, but it can actually go all the way down to 0.3 to 0.2 micrometer, which can actually get you up to the cellular lacunae canaliculi visualizations. But just so to show you here, you can see that in the younger control sample, there's not a lot of, the porosity looks normal, quote unquote, where in the older model, you can see that increased porosity and the thickening of the low mineralized layer as we typically observe in osteoarthritis model. But then how do we put a numbers to these things? So what I did then is to, the result is to show that, yes, when we separate the cortical region, shown here on the right, into low and high mineralized region, red and gray, we can see that the low mineralized area can take up to half of the cortical region that we are acquiring, which we wouldn't be able to see when we are acquiring with micro CT. And this is consistent for both older samples and younger samples as well. This ratio is higher than 50% for both groups. Then if we can separate them in terms of porosity, then you see that when I look at them combined, as we would see in micro CT, combined cortical region, the porosity looks similar in the younger cohorts. That difference starts to come to the older cohorts on the right. However, if we split them into low mineral, you can see that in the low mineral tissue, there is actually a jump in porosity all the way at the younger cohorts compared to the older ones. And likewise, the higher mineralized region tends to be more dense and thus is reflected in this area with lower porosity. And the next result that I wanted to highlight again, we can observe the same thing for thickness, as that, okay, if we look at them as combined region, as we would see in a regular CT, micro CT, sorry, then we can see that in the lower mineralized region actually has a significantly lower reduced thickness compared to the higher pair. And that kind of makes sense because thickness is kind of inversely proportional to porosity. So all those extra pores and all those rugged surfaces at the calcified cartilage interface actually plays into decreased thickness that we see in the lower mineralized tissue. Just a rough quick conclusion is that SR micro CT, synchrotron radiation micro CT with monochromatic beams and high resolution allows visualization and quantitative assessment of calcified cartilage and other surrounding cortical regions in mice. And other tissues, really. The image I showed on the cover by Dr. Shiba was actually from a human biopsy sample. And our contrast agent protocol has shown that we can identify the subchondral area, that is the region immediately underneath the cartilage, for analysis. And a significant part of the cortical region can consist of highly porous but low mineralized tissue, which we should take into account, even though we cannot see them in micro CT, but it's to be aware of this difference level in mineralization. And with that, I would like to acknowledge my colleagues here at Melbourne and at ETH Zurich. And thank you very much for everyone. Please feel free to reach out.
Thank you so much. Does anyone have any questions for Paul Pat? I don't want to ask one because this is very far from my field and I don't want to sound very silly. But I'm really fascinated with the way the level of detail that this can get. A very, very basic question, but how long did the synchrotron scan process, how long does the imaging process actually take? Like what's kind of like a day in the life of a synchrotron, if you will?
No, that's a really good question. So what the samples we use for the synchrotron study, generally we just do this on the side of the main study that we're doing. As in, while we collect samples, we analyze them, we scan them with regular micro CT, then we kind of just split the samples part for histology, and then the other groups we then, okay, let's give it a go, see what we can get out of this. Acquiring the actual image is quite fast because it depends on the resolution as well. How small do you want to go? And also, as you know, the more resolution you get, the more images you need to take of the same picture, like it's very zoomed in, so to say. So if you want to cover more of that bone, then you need to scan more time. Generally, for the images that we take, that I showed you in this presentation, like these guys here, that's like half an hour.
Nice, yeah.
Yeah. And it's you can finish everything in a day. And if the facility is generous, they can give you multiple days. So what happened was so in this study at Switzerland, we went there multiple times, multiple days, because then, it's like a scheduling system. You need to book a queue. But the one in Melbourne gave the people at Melbourne Synchrotron gave us like 5 days in a row. So we actually camped out all night.
Oh, that's great.
Scanning everything all at once. A fun team activity, a fun team building activity, I have to say.
Are there any other questions?
Yes, can I ask a quick question? Can you hear me?
Yes.
Yeah, I just I may miss the first part of your talk. It was great, but I just wonder, how did you prepare your sample for synchrotron imaging? Is the same way you prepare for micro CT imaging?
So yes.
Yeah.
Sorry, I cut you off, but yes, you prepare the sample pretty much the same way as the micro CT imaging. Say if your sample of interest is just bone and you don't really care much about the cartilage, it can go in as it is, imaged in air. They have a small pedestal that you can just attach your sample to, and then it would just rotate just like a regular CT. But the reason why we did all this fancy thing with contrastation and humidity chamber is because we want to also see the cartilage, which is typically generally not visualizable in CT. And when you start frying them with high energy radiation, it starts to dry. So it comes out looking like fried chicken bone. But yeah, so this is all to prevent that from happening. But technically, if you're just looking at bone, it's a lot more simpler. You can just go from animals and chuck them directly inside.
I see. I just wonder if you just slice the bone, it may enhance the accuracy of mineralization, do you think? Or just whole bone as it is?
I think whole bone should work because the X-ray radiation penetrates through the sample anyway. I think actually, if anything, from experience, slicing the bone actually introduced artifacts. Since the resolution is so high, any small pieces of the bone that you cut could be left inside, and then it's a question of whether is this natural bone or fragments, or it's because we just cut them.
So spread all over the place, maybe.
Yeah, exactly.
Okay, great. Thank you so much.
I had one other question pop out, which was from Pravir, just asked, is the Hexabricks contrast agent commercially available, or is that something?
So Hexabricks used to be commercially available. It's actually a human contrast agent for MRI. It's ionic, so it's great for cartilage visualization. However, it has been pulled off the shelf because people have been getting allergic reactions to it.
Okay.
Yeah, so it's not commercially available now, unfortunately. But we can have a chat on the sideline if you want about alternatives like CFO Plus or Dotarim. We've been working with some of the MRI contrasts as well, and some of them seem to work for CT.
Okay, amazing.
Yeah.
I'll direct Pravir towards you. Otherwise, I think you're off the hook. Thank you so much.
Thank you so much.
And we'll move on to our final talk for the webinar. So I'm excited to introduce Dr. Pravir Sahota, who's a biomedical engineer by training. He completed his PhD at the Indian Institute of Technology in Ropar, India, and following his PhD, Dr. Sahota was awarded the prestigious Alexander von Humboldt Fellowship and joined the Department of Osteology and Biomechanics at the University Medical Center in Hamburg-Eppendorf in Germany. Currently, he's working with Professor Buse's group, focusing on the effects of glucocorticoids on bone quality using human biopsies. He's a lifetime member of the Indian Society for Bone and Mineral Research and an active member of the International Federation of Musculoskeletal Research Societies. And his talk is entitled Type 2 Diabetes and Bone Fragility, a Multifactorial Perspective Beyond Strength. Thanks, Pravir.
Yeah, so thank you. Am I audible? Am I audible?
Yes, yeah.
Okay, so thank you for nice introduction. So the bone fragility is a big concern in diabetic patients. And to know the patients who are fracture prone, the gold standard tool is dual energy X-ray absorptiometry, which gives aerial bone mineral density. And this aerial bone mineral density is directly associated with bone strength. But despite of normal or slightly higher aerial bone mineral density, a minor trauma or side fall could lead to fragility fracture in diabetes patients. So research question is, why do some diabetic patients with similar BMD break bones easily while others don't? And for that, we hypothesize that probably the bone quality playing a dominant role here, which is not adequate, and it leads to fragility fracture in diabetes. So what is bone quality? In 2003, first time the bone quality is defined as the sum total characteristics of bone that influence bone's resistance to fracture. We have elaborated what is sum total characteristics. So it includes bone microstructure, mechanical and material property, microdamage accumulation in bone matrix. If you go to compositional aspect, then mineral content, crystallinity, crystal size, collagen content, collagen cross-linking, orientation of collagen, water content, of course, cellular activity and dynamics. And in case of diabetes, accumulation of advanced glycation end product is also playing important role in bone quality. So individual constituent plays an important role to maintain overall structural integrity of bone. And there are various techniques available in bone research field to study multiscale bone quality. Some of them are summarized here. And I have used some technique, for example, mechanical property, where we can do the compression test or bending or tensile test. Microstructural analysis can be done in 2D with histomorphometry or in 3D with microCT. Material property can be tested with nano indentation, micro indentation, and nowadays it can also be possible in vivo material property with osteoprobe. Compositional analysis with spectroscopic technique, for example, FTIR, Fourier infrared spectroscopy, or Raman spectroscopy. Mineral distribution we can study with QBEI, quantitative backscattered electron imaging, where we can see the concentration of calcium over bone surface. Likewise, there are other techniques available. So I want to come to the study that I have conducted. For that, we have collected patients who underwent hip replacement surgery following first hip fragility fracture with and without type 2 diabetes. So our main cohort include 40 number in non-diabetes group and 30 in diabetes group. And baseline demographic, you can see all is comparable except HbA1c, which is significantly high. And DEXA parameters are also comparable between the groups. A sub-study is done on cortical bone, where we included 25 cases from non-diabetes group and 10 from diabetes group. And this subset coming from the main cohort, and reason of less number of bone samples are because we did not find cortical bone intact from inferomedial region. That was reason of interest for complete complete cohort. So therefore, the subset is studied here. Both the studies are now accepted. So one is published in 21 and another is recently accepted in Bone Journal. So how we allocated the bone specimen for multi-scale bone quality analysis, 3 to 4 trabecular bone core were extracted from femoral head, and 1 bone core was dedicated for micro CT analysis for measuring microstructure. And then we tested with compression test. The second bone core we used for small scale analysis, where we studied the FAGE assay, that is fluorescent advanced glycation end product. FTIR in ATR mode for local bone composition, and gravimetric analysis is done for bulk bone compositional analysis. And then the bone core is embedded, polished, and used for material testing. And third bone core is grounded in a fine powder, and then crystal structure is, crystal size is studied. Same is done with cortical bone analysis, microstructure with micro CT, compression test, material property, and small scale compositional analysis and FAGE assay. So results says that we have low bone volume fraction in trabecular bone because of lower trabecular thickness and trabecular number. And we found increased cortical porosity in diabetes cohort. So overall compromised cortical and trabecular bone microstructure in type 2 diabetes. Mechanical property test that is done with compression says that the pre-yield property, so we use typical graph of stress strain curve, and the pre-yield properties are determined below yield point, and above yield point, all post-yield properties are determined. It's a standard way to determine mechanical properties. And we found both pre and post-yield properties are lower in diabetes group. Whereas in cortical bone, we did not find difference in modulus or yield stress, but ultimate stress, strain, and energy-related parameters are lower in diabetes group. Material property has been investigated for both trabecular and cortical bone. We found lower material property for diabetes group, whereas in cortical bone, there is no significant difference in material property. Bone compositional analysis with the spectroscopic technique. This is a standard parameter, that is mineral to matrix ratio is a relative mineral to matrix content. And it is lower in trabecular bone, whereas in cortical bone, the mineral to matrix ratio is not significantly different. And bulk bone composition is done with gravimetric analysis, where we heat the bone and we measure the decomposition of the bone at different temperature. And it is it's like a really quantitative approach. And here we found both mineral to matrix ratio for trabecular and cortical bone is lower in diabetes group. Then, of course, in type 2 diabetes, glycation in bone matrix is also important. So for that, we did glycation quantification in 2 ways. One is FAGE assay, that is fluorescent advanced glycation end product. So basically the advanced product which make cross-linking with collagen matrix has been studied. And we found FAGE content is higher in diabetes group for both trabecular and cortical bone, and non-enzymatic cross-link, which is studied with spectroscopic technique, is also elevated in diabetes group. And then we have we found a causal relationship between glycation parameter and mechanical property. So when the mechanical properties are plotted against glycation content, we found a negative association of mechanical property with glycation content. So with increased glycation content, the mechanical properties, mainly the post-yield properties, so post-yield strain energy, toughness, and post-yield strain are lower or reducing for both trabecular and cortical bone. For trabecular bone, post-yield energy, toughness, and post-yield strain are decreasing with increasing glycation content, and in cortical bone, with increasing enzymatic cross-link ratio and FAGE content, ultimate stress and ultimate strain are reducing respectively. And then we run ANCOVA analysis only for trabecular bone, because our mechanical properties were coming correlated with bone volume fraction. And we want to make sure that whether reduced mechanical properties are coming because of the effect of glycation or because of low BV by TV. And when we adjusted the effect of BV by TV from mechanical property, we found that mineral governing properties are no more significant in diabetes group, whereas the collagen governing properties, post-yield energy and toughness, are lower in diabetes group. So it confirms that the degradation in the post-yield property are not because of the low BV by TV, whereas it is because of the increased glycation. With this, I would like to summarize that we found lower BV by TV for trabecular bone, increased cortical porosity, lower post-yield mechanical properties and mineral to matrix ratio, as well as we found increased glycation content in terms of non-enzymatic cross-link ratio and FAGE for both trabecular and cortical bone. Another thing, the mineral governing parameters do not play a dominant role in diabetic bone fractures, whereas collagen dominating properties are impaired and leads to fragility fracture in diabetes cohort. Cross-linking AGE contribute to degradation of post-yield properties in type 2 diabetes. And if I want to conclude in one line, I would say the increased AGE in bone matrix leads to impaired post-yield properties, and this leads to reduced fracture resistance and increased fracture risk in diabetes cohort. With this, I would like to thank you everyone for listening my talk, and I would like to acknowledge many people who helped me, all either directly or indirectly, and also my supervisors, funding agency, and thank you. I'm open for question now.
Thanks, Pravir. Are there any comments or questions? I know we're running a bit over time, so I want to try and keep things rolling. I do have one, might be a bit of a tricky question, but so your samples are from non-diabetic and diabetic patients, but all of the patients had a fracture, is that correct?
Yes.
Yeah. Do you have plans to look at a non-fracture control group to look and see those differences between people who've remained fracture-free regardless of diabetes?
Yes, this is very relevant and very important questions. But so our aim was to see when the patient really get fracture with type 2 diabetes, what happened in multi-scale bone quality parameters. And getting the bone biopsies in diabetes cohort, those are not fracture, is really tricky. We are in line to arrange this type of biopsies, but still not successful. Yeah, so hopefully in future we would like to do that.
Nice, thank you. Any
Thank you to
other
you.
any other questions? I might just pass back to you, Kyungmin.
Thank you, Dr. Anderson, and thank you, Shiho talk. Very interesting talk, and thank you for our audience and also for great speakers from each society and also committee member to put their effort and time to invite a good speaker and organizing this good webinar from KSBMR, JSBMR, ANZBMS, ISBMR, ICMRS. I hope we held this meaningful symposium at least once a year. So I hope we can meet and talk another coming very recent research from all over you next the end of the year. Okay, and thank you for all and looking forward to meeting you in the next meeting. Okay, thank you. Bye.
Bye, guys.